1 second, we're going to set everything up. All right, the paper week continues. We're going to be reading a ton of papers this week. And today, we're going to be reading Seen Beyond the Brain, Conditional Diffusion Models with Sparse Masked Modeling for Vision Decoding. So this is a really cool paper where they are taking fMRIs, which is magnetic resonance imaging. It's a way to look at someone's brain while they're using it. And they're using it to reconstruct the image that the human being is seeing. It's quite crazy to think about. Yeah, that's what the paper is. I don't know much about it beforehand. I'm not really knowledgeable about fMRI either, so curious to see what we learn. All right. So this comes out of Singapore National University, Chinese University at Hong Kong, and then Stanford. And it's a relatively recent paper, but not in November. Not extremely fresh, just a little bit fresh. Okay, so we have this beautiful visualization here in the front page here. For the first time, our proposed MindViz is capable of decoding fMRI-based activities and decoding fMRI-based brain activities and reconstructing images with not only plausible details but also accurate semantics and image features, outperforming previous approaches. Comparison with benchmarks, write more construction examples. Okay, so this is the image that the person was seeing at the top, and then this reconstructed image is what their diffusion model generated. They're probably going to be messing with the context, where the context for the diffusion model is going to be the fMRI, but I wonder if the fMRI itself is going to have a variety of kind of feature encoding layers added to it. Deepen our understanding of the human visual system. Right, this is kind of the brain-computer interface, things like Neuralink, but also more, I don't know if I say realistic, but Neuralink is pretty far out. That's where you actually use robotic surgery to like put electrodes inside the brain. But there's a variety of consumer products that you can buy right now that just measure the electrical signal on the outside of your brain, and you can even control VR games that way. Okay, so sparse masked brain modeling with double conditioned latent diffusion model for human vision decoding. So quite a lot of words there. Okay, first we learn an effective self-supervised representation of MRI data using mask modeling in a large latent space inspired by the sparse coding of information in the primary visual cortex. Okay, so they are encoding this fMRI data, and they're using a self-supervised learning objective to build this embedding space. That could almost be an entire paper by itself, to be honest. Okay, then they get a diffusion model, latent diffusion model. So it's diffusing in the latent space with double conditioning. Okay, so there's two things that it's being conditioned on. We benchmark our model qualitatively and quantitatively. Outperformed state-of-the-art in semantic mapping and generation quality with Frechet inception distance. Exhaustive ablation study was also conducted. So I mean, it's interesting that FID here, we're in the paper yesterday, there was a section that talked about how FID might be wrong or not appropriate in some contexts, so we'll see if this context is one of those. What you think is what you see. Open up with a nice quote. Perception of the world is determined not only by objective stimuli, but also by our experiences. True. Yeah, non-invasive. I think that's the key word there, right? There's not putting functional magnetic resonance imaging, fMRI. You sit in basically this giant toroidal magnet, and then the magnet measures basically the spin of the electrons. I want to say it's the electrons, but it might be the protons. But it measures the spin of them, right? Because particles have a magnetic spin, and there's a resonance, right? So like when you have specific frequencies of the changing magnetic field, you get like a resonance. I'm not exactly sure. How do MRIs work? MRIs employ powerful magnets which produce a strong magnetic field that forces the protons, okay, so it's not the electrons, in the body to align with that field. When the radio frequency current is then pulsed through the patient, the protons are stimulated and spin out of equilibrium, straining against the pull of the magnetic field. Okay, and then you can measure that and somehow localize each of those protons. With the help of recent deep learning, blah, blah, blah. There isn't fMRI image pairs. Reconstructed images are usually blurry and semantically meaningless. I would say we do have some understanding about what the different areas of the brain are, but I agree that when it comes to the resolution required to know exactly what you're looking at, that, yeah, we generally don't have a ton of meaning there. Individual variability in brain representations further complicates this problem. Powerful brain decoding regions should robustly recognize features shared across a population over a background of individual variation. I think this is an interesting take here, and this figure kind of shows it, right? There's going to be a variety between different humans when it comes to the brain activation in what they call here regions of interest activating during the same visual task. And I think there's actually super cool stuff in neuroscience with, for example, blind people. Blind people, right, they don't have eyes, so there's this whole area of their brain, the visual cortex, that in theory would be unused, but the brain actually is very smart, and it learns to reuse that part of the brain for potentially hearing or some other kind of sense, and it augments that sensor, that the hearing of someone who's blind by using that part of the brain. So I imagine there's like less extreme versions of that happening in humans that have vision, and humans that have slightly different shaped brains, slightly different kind of biases towards hearing or biases towards vision. So you're going to have different activations in different humans. From a large-scale data set with rich demographic compositions and relax the direct generation Conditional synthesis allowing for sampling variants under the same semantic category. Self-supervised learning, I think that's a great way to try to get a embedding space for fMRI images. Yeah, so people usually do this with something like classification, right? So they'll come up with some classification task and then use supervised learning to train it. But I think that that's kind of what you would do 10 years ago. I think nowadays, maybe not 10 years ago, maybe 5 years ago. Nowadays, with the kind of size of networks you have available, the availability of compute, the fact that you can train for longer, I think these self-supervised tasks, especially if you have a huge amount of unlabeled data and a very small amount of labeled data, unsupervised and self-supervised learning are going to be the way to go. Okay, so the specific self-supervised objective they're going to use here is masked signal modeling, MSM. Okay. Okay, so there is some other work here that has done some brain decoding, but they're saying they use a much larger representation to data space ratio to boost the information capacity of the learned representations. And this is something that we kept repeating yesterday and over the course of these various diffusion papers that we've been reading. But the quality of the representation, or that like feature vector that you're using to as the context to a diffusion model, the quality and the richness of that embedding space is going to be very important to the quality of the output of your diffusion model. Proposed sparse coded, blah, blah, blah. Yep. This is the unsupervised learning objective. Augmenting latent diffusion model with double conditioning. Okay. Integrating the representation ability with the generation ability. Generates more plausible images with better preserved semantic information. Okay, so generates better. Quantitative and qualitative tests are performed on multiple data sets, including a new data set that is not. Okay, so there's a new data set. Okay, so I mean, this is a pretty big paper, lots of different stuff in here. They have this SCMBM, this masked modeling that they're using to create a kind of feature space for fMRI images. They're also using that feature space to provide context to a latent diffusion model with this double conditioning that they're doing. Their result with that is not only kind of cool images that are subjectively better, but then they also create a new data set here to kind of benchmark their approach to other previous approaches and then set the stage for people to come and benchmark on this new data set as well. Okay, so conventional decoding methods. Rely on training and corresponding hierarchical image features extracted by a pre-trained VGG. Yeah, so this is something you could do, right? You could take the images here and then just feed them through an old ConvNet that's been pre-trained on ImageNet. But the ConvNet trained on ImageNet, such as a VGG, like that's not gonna really have any brain specific like knowledge encoded inside that representation space, right? It's really just going to be more of an edge detector and a texture detector and a variety of different like useful kind of feature maps that it convolves around the image, but none of them are really related to fMRI. So even though it's better than nothing, I would say that it's probably not going to be anywhere near as good as what you get if you do this masked signal modeling. Okay, well, maybe they say here you can just use an autoencoder. Maybe that's a different way of getting the image or the embedding space. Okay, so people have done this for with GANs, basically. Just you train a regression model to extract a latent fMRI representation, and then you fine-tune a pre-trained big GAN that's also conditional, so you can give it some vector to generate, some vector to condition the generation of the GAN. Okay, so that's kind of cool. So basically, these two papers here, 32 and 34, they effectively did what this paper is doing, except with GANs rather than diffusion models, and then a slightly different way to create this representation space. MSM is a generalized denoising autoencoder that aims to recover the original data from the remaining after masking. So that's the unsupervised objective, is you're masking parts of the image. So you might have maybe this image, you mask out a part of it, and then you put it through an autoencoder, and then you try to get it to reproduce the original image. So that's what it means by self-supervised learning, right? In supervised learning, you are training something like a classifier. You know exactly what the input and the output of the network should be, which allows you to get a gradient, which allows you to train that network over time. But in supervised learning, you need this, every single input needs to have its matching output. Self-supervised learning, you're creating the input and output by, for example, here, masking. So you don't have the label for the image. You don't actually know what that image contains, but you can mask out part of it and then try to recreate the full image. And in a way, it's like supervised learning, but because you're creating that supervision task kind of from thin air, that's where the self part of the self-supervised learning comes from. Okay, so there's extremely high mask ratio of 75% and low mask ratio of 25%. So in an image, for example, you would mask out 75% of the image, but in something like a language model, you see a lower amount of masking, something more like 25%. Okay, let's see how they define diffusion models here. So diffusion model is a probabilistic model defined by a bidirectional Markov chain of states. Sure. So bidirectional means you can go both ways, right? Markov is a Markov process where you have a certain state, you take a certain action, you end up in a new state. The Markov process is used in reinforcement learning a lot. But it's basically just kind of a graph. You could think of it that way. There's kind of nodes, and then you can take actions, which are the edges. Okay, so the better mental model to think about is that you have a diffusion process that gradually adds noise to the data. And then you have a reverse process which recovers the corrupted data by modeling the posterior distribution. Right, so you have a You're gradually adding noise, and then you're removing that noise because you know the noise that you added. T is the number of steps that you're going to take in terms of adding noise and removing noise. So in other diffusion papers, they might call this the number of samples. But here they basically are saying that it's the length of the Markov chain, which is another way of describing what this the total number T, big T is, right? T total steps of denoising or noising. You use Gaussian noise here. I kind of like that the wording for this is different than the wording we've seen in a bunch of other diffusion papers, right? Here you have a lot of, I like the wording here because it kind of makes you realize statistically what you're doing, right? Where really what you're trying to learn is a conditional probability, right? You have the image at the time step previous conditioned on the current image, right? That's a conditional probability. A reverse conditional probability because you're moving backwards in this time t. Or I don't want to call it time t, but they gave it the letter t, so I can't help it. All right, and then you are ultimately, the loss here is a variational lower bound, right? So it's the expected value of the image x and the noise sigma here. The noise itself is drawn from a normal distribution centered around 0 with a mean of 1, right? That's Gaussian noise. And the expected value of that at some time t, and the expected value is of this term here, which is the L2 loss, right? So the absolute value squared of the difference between this noise and this noise here. Oh shit. And that is the variational lower bound, what they call simple, so the simple loss. Where here is a set of denoising functions. Okay. So latent diffusion model, this is also part of the related work here. Apart from the conventional diffusion models that generate samples in the original data space, right? That's what a normal diffusion model be. But there's another category of diffusion models that generate samples in the latent space. So rather than adding noise to a image as you have here, you're adding noise to the latent vector itself, which is going to be smaller. So it's going to be, yeah, here you go. Operating in the latent space reduces the computational cost and introduces less spatial downsampling, right? Because an image is going to be, I don't know, for example, this image of the cat, it might be 128 pixels by 128 pixels, and then there's 3 channels. But a latent vector, like this Z here, is going to be much smaller. Maybe it's 256 numbers total. Like it's like a float vector of length 256. Maybe it has a little channel or batch dimension or something like that too. But it's just a smaller amount of numbers, less matrix multiplications, less gradients to calculate. So it's easier to add the noise, remove the noise, right? Every little micro operation that you do is faster because and takes less memory because the vector is smaller than the full image. LDM proposed in 42 consists of vector quantization regularized autoencoder that compresses images into a lower dimensional latent features. Yeah, so vector quantization is when you have a embedding space, right? And it's a multi-dimensional space, right? And any point in that space is basically, you can think of it as a vector. And that vector is infinite precision, right? There's a vector at 1, 1, but then there's also you could have a vector at 1.00001, 1, right? So vector quantization is this idea where rather than kind of having like an infinite resolution within an embedding space or some kind of like high dimensional space, you can quantize it into specific areas, right? So any vector that is within some distance of 1, 1, you just say this is the same as 1, 1, and that quantization, by choosing kind of the unit that you're kind of like rounding everything to and quantizing to, you get a discrete set of values, of potential values for the embeddings, and then that helps you down the line. Okay, then they have a UNet-based denoising model. Okay, this is kind of interesting here. Incorporating attention mechanisms into the UNet allows the flexibility to condition image generation through key value query vectors during the Markov chain transitions. Okay, 3, methodology. So here they're going to go over probably what they have in figure 3 here. So why don't we read the section and then we'll go and we'll look at figure 3. Okay, so in this subsection we provide a detailed analysis of the fMRI. And elaborate on the motivations of our designs. fMRI measures the brain blood oxygen level dependent changes as 3D voxels that serve as a proxy for the underlying changes in brain activity. Okay, so fMRI is measuring, right, the magnetic spin of the protons, and what it means here by 3D voxels is that the space, the volume that your head takes up, you can break up that volume into tiny little voxels, right? Tiny little cubes that kind of represent the 3D position of that point in space. And the fMRI will tell you, will give you a number for that specific voxel in 3D space, and that number kind of represents the brain blood oxygen level dependent changes. So it's kind of a long-winded way of saying that what the fMRI can largely measure is regions in your brain that have more blood or less blood. And that's how they can tell what regions of your brain are being activated and which regions are not. Because blood flow can be used as kind of a proxy or an indication that that region of the brain is being used versus not being used. fMRI data is averaged across the time during which the stimulus is presented. Okay, so there's some like 5-second period where they show the person this image, and then they measure at whatever the frequency of this fMRI is, and then they average all of those individual snapshots over that 5-second view period, and then you get that image. Okay, so region of interest of the average data. The ROI size is generally smaller than the image size. For example, has about 45,000 voxels in the visual cortex. This creates a large difference in dimensionality when transforming fMRI into images. Okay, so the resolution of fMRIs is pretty small, is basically what you learn there. And fMRIs from different data sets may have different... Okay, yeah, so basically there's a lot of different settings with these fMRIs. So if you record in one fMRI, it's unlikely that a different fMRI will have similar looking data. It'll be different in some way. So you have this kind of problem where it's anything that you train with data from one fMRI is probably not going to generalize to a different fMRI. And that kind of sets up the stage for getting these large fMRI data sets that are collected on a variety of fMRI machines so that you can hopefully generalize to new fMRI machines. Okay, so there's two stages here, stage A and stage B. So stage A is when you are learning the embeddings, right, the representation space of this fMRI data set with this semi-supervised masked signal modeling task. And then stage B, you have your pre-trained encoder, and then your latent diffusion model. And the encoder is jointly fine-tuned with cross-attention heads in the LDM using paired annotations. Okay, that's interesting. So they're not freezing the encoder that they learned with the self-supervised signal. They're actually fine-tuning it with the diffusion model as well. So that is interesting. All right, and then they kind of talk more about stage A here and stage B. Let's look at the figure 3 here. Mass brain modeling, stage A here. So they have their input fMRI. I think it starts here. They patchify it and then mask it. So patchify, I think, is maybe some of the stuff that they're doing here, where they're basically taking this fMRI raw data and then kind of voxelizing it. And then, right, for each different little part of the brain, you're going to have a single number that represents kind of the amount of blood flow. Then, of course, you're taking the full patchified fMRI, and then you're going to mask out a certain part of it for this self-supervised learning objective. That's what they're doing here with the masking. And they're going to basically create this tokenizer, if you want to think of it that way, that can take a raw image and give you a embedding. Or maybe not. Maybe okay, this tokenizer is not what I'm talking about. What I'm talking about is this here, this blue thing called fMRI embedding. So this tokenization here is just a simple step, you could think of it just like the first part of this encoder decoder self-supervised training setup that they have here, that basically the encoder is compressing this masked brain patches into a small little latent vector, right, for the embedding or the representation. And then this decoder is blowing it back up to the final fMRI image. And you can train this encoder and decoder at the same time such that you get very good reconstructions because you know what the input was. And the latent space that results from that is going to have all that information in there about all that semantic meaning about fMRIs just kind of embedded in there, right? That's the use of the word embedding space. And then you have a latent dimension projector. Okay, maybe this is where they do the vector quantization. Not sure. I think they'll explain this better. But the output of all of this is a little vector of numbers, which they call the fMRI embedding here. So you take an input fMRI, and this whole process converts it into a little vector of numbers. So that leads us to stage B. In stage B, you're going to take your ground truth image. I don't know if you guys can see my mouse or not. Maybe not. You're going to take your ground truth image. You're going to convert it into a latent vector, right? Then you're going to be doing your adding noise to this latent vector and then denoising it, right? So the diffusion model is learning how to remove the noise. In this case, it's noise in the latent space. That's how you train this diffusion model E here, encoder. And then the decoder D here, which takes this latent vector and converts it back to an image, is how you get the generated image. Right? But so when you're actually doing this in inference, you'd get an fMRI, you'd patchify it, you'd feed it through the VIT encoder here, you'd get a latent, you'd get the latent dimension, you'd get the embedding, you'd take that embedding, and then you'd feed it here in this double conditioning setup that they call here. So they have a cross attention, and then they have a convolution plus batch norm plus rectified linear. So that's a activation function. They have a time embedding. So maybe some kind of position sinusoidal or something that represents, like you see this in language and sequence tasks where there'll be like an additional number that tells you where along the sequence, where within the sentence it was. Maybe that's what they mean by this time embedding. But both of those feed into this attention residual U-net. So a U-net is a net that's kind of really, that's very wide and deep on one end, and then it goes down and becomes very, very the opposite of wide, thin, and then it goes back to being very wide, right? So the size of the U-net is big and then small and then big. That's why they called it a U-net. So this U-net is what actually does the denoising, it seems. This is the actual quote-unquote diffusion model. And it's taking the latent at some time t and then giving you the latent at time t minus 1. So, right, at time capital T, that's when you have the maximum amount of noise. At time 0, I suppose, that's when you have no noise. So you're basically using this U-net, which is double conditioned on the fMRI embedding in these two different ways here. And that is giving you the image latent at one time step before. And then you do that for however many time steps you have here, right? And then the final output will be this latent vector Z that should have all the noise removed. And then once you have that latent vector Z, which has all the noise removed, you can just decode it into a generated image. Here. A little sip of coffee. We got espresso coffee here again. Okay. So we have here pre-train on fMRI with SMBC or SCMBM, masked brain modeling. We randomly mask the fMRI and tokenize them to large embeddings. Curious what this tokenization means. Tokenization in text means basically breaking up a sentence into these little like word pieces, and then each of those word pieces becomes a vector. But I don't know what they're tokenizing with here. We train an autoencoder here, capital sigma MBM, and decoder D. So you have autoencoder has an encoder part and a decoder D MBM to recover the masked patches, right? This is the two parts here. Stage B, integration with the LDM through double conditioning. We project the fMRI latent, right? Right here, this Z, through two paths to the LDM conditioning space via a latent dimension projector. Where is this? PFMRI to condition. Ah, here, this is the thing. Latent dimension projector. One path directs directly to the cross-attention heads. I think that's this blue one here. And then another path adds the fMRI latent to time embeddings. Okay. The LDM operates on a low-dimensional compressed version of the original image. Yep, the latent space. However, the original image is used in this. I like this picture of a cat yelling. Activity in the human brain involves nonlinear interactions amongst 86 billion neuronal cells in the brain. How many parameters in GPT? GPT-4 will have 100 trillion parameters. So, kind of goes to show you that we should be able to achieve much higher levels of intelligence at the scale that we're currently at. Right, the human brain can achieve pretty significant intelligence with orders of magnitude less parameters, in this case their cells, than GPT-4. Okay, you have voxels of fMRI data. Okay, so you can actually mask. So this is what the ground truth looks like. So this is the fMRI data, and this is like the volume of blood in a particular voxel, you could think of it. Or I think there's 4,500 voxels total. And then they're masking it, and there here you see the ground truth, how much kind of like redundant information there is, and then if you mask it, you kind of see how you can get pretty much, you can get the idea of it pretty well. So even with a high mask ratio of 0.75, they say they can still get good results. However, we use a large embedding to patch size ratio, which significantly increases the information capacity. So what do they mean by that? What they mean by that is that the embedding space, or maybe it's the token embedding, is pretty large. So you don't have this kind of like chokehold where the dimensionality of the input signal is way higher than the dimensionality of the embedding. In this case, the dimensionality of the embedding is actually even potentially bigger than the dimensionality of the input, which makes, in this case, they're saying you're getting more capacity, the latent space is bigger, there's more design space to kind of encode this information. They call this sparse coding. And they say that the brain is good for sparse coding. Brain signals are good when sparsely coded. It seems like they reference a paper there. Okay, they make the decoder small. This is the decoder here. Because they don't really care about this decoder. Like, ultimately, all you want is the encoder. So you'll see sometimes the encoder and the decoder will be basically symmetric. They're the same architecture, just kind of backwards, same size roughly. But in this case, because the encoder is much more important to them than the decoder, they just make the decoder small, and then they put all their model capacity in the encoder. All of their computational budget, if you want to think of it that way. Theoretical and empirical studies suggest that visual stimuli are sparsely encoded in the primary visual cortex. Visual information of natural scenes can be reconstructed from a small portion of data collected from the primary visual cortex. Each patch is encoded into a higher dimensional vector space with a size much larger than the original data space. Okay, so the high dimensional embedding compared to the input size, that's this part here, this token embedding. Okay, and then in stage B, to further decode visual contents from this abstract representation, we formulate the decoding task as a conditional synthesis problem and approach it with a pre-trained latent diffusion model. Yep. You're synthesizing something while conditioning on something, right? And in this case, you're synthesizing latent vectors, and then you're conditioning on these fMRI vectors or embeddings. Everything is a vector, and embedding and representation and encoder space, like there's a lot of words in machine learning that kind of mean the same thing, and a lot of overlap between terminology. So I apologize if I'm sometimes using a word to mean something, and then later I use a different word to mean that same thing, and it can be a little bit confusing, but once you kind of realize that a lot of things mean the same thing, I think machine learning becomes easier, or at least understanding these type of papers. So the LDM operates on the image latent space, okay, which is they're doing here, sigma x. X is an image in pixel space. E of that is a vector quantized encoder. In our setting, we omit. Okay, so this is the where the vector quantization happens here. It's in this encoder and this decoder. That's quite clever. It's clever for reasons that we'll see later. The use of a vector quantized encoder. In our setting, we omit the encoder and use X directly to represent the latent variable for simplicity. Specifically given in the fMRI data Z, this is the raw data, we aim to learn the reverse diffusion process formulated by Q of this, right? So your diffusion process, the reverse diffusion process, diffusion is adding noise, denoising, or reverse diffusion is removing the noise. And it gets you the image at time t-1 conditioned on the current image and then the latent z. But then again, the X, I'm just gonna keep saying image, but in this case, remember, it's not the actual image, it's a latent vector, which is then converted into an image. Conditional information is applied through cross-attention heads and the attention-based unit. Cross-attention, of course, if you're familiar with transformers, there's these kind of three important concepts there: the queries, the keys, and the values, right? This is basically just the equation of transformers. This is how like attention heads work. And these are the matrices. So the query weights, the key weights, and the value weights, and these are what you're learning. So these are the values that are the weights and biases of the neural network that you're updating with your gradients and slowly learning over time. Diversity and consistency. Yeah, there's kind of a trade-off there. Making a point here that fMRI to image transition relies more on generation consistency. Decoded images from similar brain activities are expected to be semantically similar. In this way, we integrate the cross-attention conditioning called time step. Okay, this is the time embedding stuff that they're adding here. In time steps conditioning, we add sigma theta, tau theta to time step embeddings, where sigma theta is another suitable dimension projector. Time step embeddings are used in intermediate layers of the unit. Thus, we have phi I of x of t is phi I of x of t sigma t zeta tau z. We further reformulate the optimization to a double conditioning alternation. Oh wow, I can change the color of the highlight. Kind of cool. Okay, so this is the original kind of loss from above up here. But now, rather than, they're basically adding additional terms. So rather than having this term here, you see how now the term has been updated and it's a more complicated function. Now, a function of not just the image and the time, but also this tau and sigma of tau, which is the time embedding. Time embedding dimension. I'm still not 100% sure what this is doing. Right, like they're taking this latent vector, and I feel like you could have just done this blue arrow, this blue path here, and be fine. Like, I don't know why they had to add this pink path. Right, you have now this additional pink path that takes that this latent vector here, the fMRI embedding, and then puts it through an additional network here that also needs to be trained and also is parameterized, right? They say here parameterized by theta. It seems like kind of convoluted. Maybe they found that it gives you good results. After it's pre-trained, it's integrated with the pre-trained LDM through double conditioning. Commonly, the encoder's output is averaged, or a class token is appended to produce a pooled 1D feature vector for downstream task. we used convolution layers to pool the encoder's output into a latent dimension. There are so many encoders here that you can't just say encoder and expect people to know what the hell it means, right? There's this tokenizer here, which you could describe as an encoder. There is the encoder here in the masked brain modeling, which you could say is also an encoder. There is the encoder here, which goes from the image to the latent. And then you also have this encoder here, the time embedding, which encodes into this, from this latent into their time embedding. So you have like 4 different encoders here. Some confusing shit. Fine-tuning the cross-attention heads is critical for bridging the pre-trained conditioning space and fMRI latent space. The fine-tuning is performed end-to-end with fMRI image pairs, during which a clear connection is learned. Okay, so 3 datasets were used in this study. Human Connectome Project, HCP 1200. The Generic Object Decoding Dataset, otherwise known as GOD. That's quite funny. And then Brain Object Landscape Dataset, known as Bold 5000. So 3 different datasets here. These all seem like small ones. This is likely at 1,200. Actually, here we go. Combining these two, so they have a total of 136,000 fMRI segments, which is roughly 340 hours of fMRI scans. Okay, so in terms of the images that people are seeing, you have 1,250 from 200 distinct classes. This is interesting. The training set and the testing set have no overlapping classes. The fMRI pre-training model is similar to VIT large. So this is a vision transformer, where basically, you take an image and you break it into these patches, and then you use a transformer to look at those patches as if they were little word tokens. And it's a common kind of image encoder nowadays. It's kind of replaced the convolutional neural net as the go-to encoder. The patch size of 16 is the default patch size for that. 1024. These numbers seem familiar, so I think they're just using the VIT large kind of its default setup. They're not kind of tuning too many of those hyperparameters there. They have their mask ratio of 0.75. Training for 500 epochs, fine-tuning for another 500. Images are generated at 256 by 256. Okay. Following 16, we use the N-way top 1 and top 5 accuracy classification tasks. So top 1, top 5, again, it's a way of measuring accuracy in computer vision and in classification tasks where you don't need to necessarily get the answer correct, as long as you basically the model gives you the top 5 possible choices. So you might see this picture of a people on a boat, and it might tell you, okay, this is a boat or people or river or tree or water or something. And as long as the actual answer, which in this case might be people, is one of the top 5 answers, it counts. So that's why top 1 versus top 5. Top 1, very stringent, has to be the highest category. Top 5 is as long as you're in the top 5, you're fine. Okay, so they use a pre-trained ImageNet classifier to determine the semantic correctness of generated images. And here's your famous Fréchet distance. Fréchet. Training and testing were same subject, as individual differences remain a barrier when decoding at the group level. They compare to a couple different approaches here. Our method generated plausible details such as water and waves, which were not present in the previously coded images. This is interesting here. So color is sometimes mismatched, but one of the ways that they say that this is happening is that the color category information is processed in the frontal lobes as a cognitive process, while the visual cortex only recognizes the differences in color. Right, so all of this work here, they're only looking at the patchification of the back part of the brain, right? The visual cortex, which is in the back of your head. But what they're saying is that color information doesn't just come from the back of your head, it actually comes from the frontal lobes here, prefrontal cortex, which is more, you can think of it like the reinforcement learning part of your brain that makes those kinds of decisions, which is interesting to think about. Okay, coding at different random states. Okay, let's actually look at these results. Images generated by our method were consistent across different sampling trials, sharing similar low-level features and semantic. Okay, so this is the ground truth image, and then this is the reconstructed samples. So I think this is where it's important to talk about the fact that this encoder here is quantized, right? So they're not doing any of this in image space. They're doing it all inside this latent space, Z, right? But this encoder and decoder, which go from the latent space to the image space, are quantized. So what that means is that this encoder and decoder, right, like this vector Z, it's not telling it, it doesn't contain the information of like a fire truck with two, like it doesn't have a very nuanced representation of it, right? Because it's quantized, really you can almost think of it like it's largely just saying a fire truck from this rough view with this rough size, right? So that quantization there is kind of a trick if you want to think of it, because it takes any kind of sloppiness that could be the result of this denoising, and then just kind of like gets rid of some of that sloppiness by quantizing it and then feeding it through the decoder here. It's a terrible explanation, but I think that's one of the reasons why their images look so clear, you know. Here they're comparing to different methods here. So ground truth here on the top, this is what the person actually saw, and then this is what different methods were able to decode from the fMRI images. So the fMRI images goes all the way through that pipeline and then outputs the little latent vector, and then the decoder takes that latent vector, turns it into an image. So you can see that the category is always kind of correct, right? Lizard, lizard, building, building, water orca, water orca. So like, but for example here, this one is wrong here. You have bowling ball and it's just something circular. In this case, like a pressure gauge. But overall, their results do look a lot better than the previous results. Okay, here they have some ablation study. Okay, so this is without the masked brain modeling, which is without the pre-trained task that creates this kind of embedding space for the image, for the fMRI images. And you see how the accuracy is terrible. So that pre-training task to create a kind of rich representation space, rich embedding space upon which you will project each fMRI image is very important, and that's kind of what they're showing you here, where the accuracy is very low. And why are the parameters so much higher? And the reason the parameters are so much higher is I think because the dimensionality of the input is probably much higher if you don't encode it. Yeah, without SCMBM, these two models even failed to generate sensible images. Okay. Patch embedding dimension. Boosting the fMRI representation size using a large patch embedding matches the sparse coding mechanism. This is kind of like a hand wavy reason for that. I don't know if I buy that reason. Maybe the brain does some kind of sparse coding, but using that as a reasoning to put sparse coding in a tiny part of this giant pipeline, a little stretchy, but different patch embeddings. Okay, so this is the embedding dimension here, right? So you can see how with a small embedding dimension, you have worse performance than a larger embedding dimension here, right? And then the third kind of ablation that they have on this table is the mask ratio. So how much are they masking that input? And I think they kept saying 0.75 is what they used here, but here in this particular ablation study, it seems like somewhere between kind of 0.45 and 0.65 is kind of the sweet spot. Okay, this is the double conditioned latent diffusion model. Notes on that design and the ablations on that design. Okay, so here's where you have the double conditioning on either just the latent or the time embedding as well. So without the time embedding, honestly, that's pretty good. Cross attention condition and time condition. Yeah, honestly, this score is pretty good. I don't know why they're complaining about that. Oh, okay, I know why, because they're comparing it to here. Okay. E is the encoder. A is the cross attention heads. Okay, you're fine tuning either the encoder only or the encoder and the attention heads. And your pre-trained latent diffusion model label to image or text. Okay, so they are using a pre-trained latent diffusion model, and there's a couple different types here. They have an ImageNet 1. They have a Layon 1. This is what they're calling label conditioning, text conditioning, and then they have open images 1. They're calling layout conditioning. And it seems like the image 1 is the best, so the ImageNet 1. Sense, I think Layon and open images are more less diverse images. Let's see, I don't even know. Okay, actually, Layon and open do have diverse data sets or diverse images. We attribute the main reason to their poor performance to the complexity of their conditioning latent space. With limited training pairs, the class conditioning latent space is easier to adapt to compared to the latent space. Yeah, so this is an interesting point, right? Where if you're using a pre-trained model that was trained with a kind of class classification task, that resulting feature space or representation space or embedding space or latent space is going to be separated out so that different classes are grouped together, right? All the dogs are going to be grouped together, all the images of dogs, all the images of people, and so on. But you're saying here that Layon and open images, right, these pre-trained LDMs were trained on more complicated things, such as text conditioning, or text and some kind of text-based thing. So in that case, the resulting latent space for those is going to be more complicated. It's not just going to be all the images of dogs next to each other. There's going to be a more complicated kind of mix of stuff there, right? Because it's trying to, like, the information that it's trying to put in that latent space is more complicated. So, I mean, it's important to realize that ultimately what this paper is doing, it's like the class, it's limited to just like, hey, you're looking at a picture of a lizard, and we can tell you're looking at a picture of a lizard, but it's not like really understanding deeply what your brain is doing in terms of like the edges and like the way that you're seeing it, and like the green color, maybe the contrast. Like there's so much more in this image that your brain is recognizing, versus like ultimately what this entire paper is just getting from this, and this entire approach is that you're looking at a lizard, and here we're going to generate a picture of a lizard for you. So it's really it's simpler than it seems. Still super impressive, but simpler than it seems. Okay, so they validate with bold 5000, different data set here, bold 500. Okay. I think this is a mistype. It should be bold 5000. Image reconstruction quality varied across subjects. So you're saying here that people might be thinking about different things. You show them an image, but you show them an image of a dog, and then they think about their dog at home, and then they're mostly thinking about their house rather than the dog. So it's difficult to really know for sure what someone's thinking about. Okay. Scroll down to the appendix. See, there's some interesting stuff there. Oh, huge tables. 250 steps. So that's the capital T in the equations before. Pretty good here. Some mistakes, for example. Here you have a picture of a plane, and people somehow get a picture of it like a goat. And then here you have a picture of kites in the sky, and it gives you the plane. A lot of these are pretty good. Dog, badger, weird animal. Samples for ablation study. Okay. fMRI pre-processing details. Signal to noise ratio, a higher signal to noise ratio. Okay, so they had 5 different people total. And you can see how for some people the accuracy was way higher than for others. I think that probably just means that the number of test images was small. If you just get 1 or 2 wrong, you're screwed, kind of thing. Pre-training, fine-tuning. Okay. That is a dense paper. So this was Seeing Beyond the Brain, Conditional Diffusion Model with Sparse Masked Modeling for Vision Decoding. Very interesting work, thought-provoking. I think it's somewhat over-engineered. There's quite complicated amount of crap here. Like, this would have been cooler if they got good results without all this kind of extra stuff that they did with this double conditioning, the tokenization, the masking, this UNet, this time embedding, doing it in the latent space rather than the image space. I feel like there's an opportunity here to take the data sets that they have, greatly simplify this system here, and train it for longer and see if you can get cool results. But sometimes you can't get the cool results without over-engineering, so hats off to the team here for going above and beyond and making this kind of complicated system in order to get these cool results. Yeah, very interesting. I think that at some point it'd also be cool to look at other fMRI regions, fMRI scans of other brain regions, right? Like the prefrontal cortex while you're thinking about a puzzle or something like that. Or the motor cortex while you're moving your hands, right? There's other regions of your brain that are more accessible, like the motor cortex that controls your arms and your legs and stuff is actually on the surface of your brain, kind of around the top here. And I feel like that would be a cool one. Just have people play with blocks or something, or like touch something on a screen and then decode it from their brain. Cool, that was MindViz. And yeah, we're going to continue with the Paperpalooza this week. We'll be doing another paper tomorrow and Thursday and Friday, and we'll see what comes from that. But thanks for listening, and see you guys later.